Good evening, everybody. We will be studying applications of animal tissue culture. Here, as you see, we have dissected an animal onto which we have isolated tissue and followed by separation of cells in vitro. Following this, we have cultured these cells in the incubator having tissue culture flask and tissue culture plates. So in this way, a single animal liver has been isolated and it has been grown into number of tissue culture flasks and number of tissue culture plates. So a single liver that has been isolated from the rat has been used for multiple drug concentrations to be tested at a time. In case we want to test any drug for say hepatotoxicity, we will have to give the drug to multiple animals. Instead of that, here in animal tissue culture, what we are doing is either in the primary culture, we are taking the single organ and are using it for multiple concentrations to be tested on the isolated cells. In this way, we are reducing the number of animals to be used for drug testing. Hereafter, there are number of cell lines which are available and which have made this technique very much important, and that is reducing the number of animals that are required. Here, if you are using cell lines, you do not need any animals to be tested on the cell. And also, there are number of cell lines available from the human tissues, because of which these become the tools to test the drugs in human cells itself. So now, with this background, we are going to see the applications of tissue culture in various fields of the pharmaceutical, biopharmaceutical, diagnostic, and therapeutic biopharmaceutical production and its importance in current pharmaceutical scenario. So here is the actual definition of animal tissue culture is what it's an in vitro growth and maintenance of animal cells, tissues, organs is known as animal tissue culture. Its generic term to include organs and culture the cells to obtain the in vitro sustainable long-term usage of these tissues and cells is the main motto of animal tissue culture. This technique, when we are using cells which are isolated either from the primary organ or the cell lines, they can be termed as cell culture. So the culture derived from dispersed cells taken from original tissue or from the primary culture or from a cell line or strained by enzymatic or mechanical or chemical disaggregation of the tissue are known as cell cultures. Hence, as we have discussed, the main importance of animal cell culture is we are culturing the actual life in petri dish and we are experimenting with it. In this process, we get advantage to save lives. So without sacrificing animals, we are able to test drugs directly on various type of cells. There are, there are different cell lines available from human tissue or from the cancerous or immortalized cell lines for various organisms, right from humans, rat, mice, guinea pigs, rabbits. Most of the animals which we are using for drug testing, their cell lines are available, which are saving lots of lives of animals. Now, coming to the scope of its applications. So using cell lines and culturing these cells in vitro, we can use them for studying various intracellular activities, intercellular activities, environmental interactions, cell-to-cell interactions, cell products, genetics involved, okay, and intracellular flux can be studied by these cells which we are culturing and experimenting it with. So let's check which intracellular activities can be studied. So here, intracellular activities like transcription, protein synthesis, energy metabolism, drug metabolism, gene expression, and apoptosis, all such intracellular activities can be studied by using cells in petri dishes and performing various staining techniques or various types of assays which are able to give us all the experimental results regarding intracellular activities. Intracellular flux, that is the transport in between the cell and the nucleoli. Okay, this is internal to the cells. So RNA molecules, hormones, metabolites, signal transducers, membrane trafficking, proteins, these are all studied by intracellular flux studies. Now, the environmental interaction. Your cells are growing in a different type of microenvironment, and hence, though all the cells contain the same genetic makeup, they behave differently in different tissues, and the microenvironment is responsible for the specific gene expression in that particular cell. So, ligand receptor interactions, their infections, drug actions, and interactions in the surrounding microenvironment are important to study, and such environmental interactions are very much being in a controlled fashion can be studied in the animal cell culture models. These animal cell culture models provide an added advantage that the whole environment in situ is different as compared to the controlled environment in vitro. Here all parameters are well defined, which is not possible in vivo, and hence when we want to pinpoint any mechanism of action, we can study under controlled conditions in, say, in vitro conditions by using animal cell culture techniques. Now, to study cell-cell interactions, these models can be used by using co-culture techniques. Here, you can study contact inhibition, density limitation of growth, embryonic induction, metabolic cooperation, matrix interactions, and invasions. There are various assays and staining techniques which can be employed to study cell-cell interactions. Cell products are the major upcoming field in the biopharmaceutical, where we are producing number of cells and their secretory products which can be used for therapeutic purposes. So cell products, single cell proteins are becoming the huge biopharmaceutical industry with the upcoming various monoclonal antibody production and therapeutic antibody production. Hybridoma technology is being used and it requires applications of various techniques in cell culture. Now coming to the basics of genetics, which can be studied by using these cultured cells and the primary cells. So you can do genetic analysis, you can perform experiments of genetic manipulation, interventions of gene therapy can be applied to different cells, transformation studies that how a cell gets transformed from normal cell to tumorigenic cells that can be studied by applications of animal cell culture. Now, when we want to continue to use a normal cell line which has a definite lifespan, but if we want to use the same cells for a longer duration of time, we want to keep on culture them for number of more generations, we can introduce immortalization to these normal cells. And this technique of immortalization can be applied to these cells, which provides us with the cell lines which are comparable to the normal cell lines. So you can study various phenomena on the normalized or immortalized cells which are near about to the normal cells. Going further, we are now going to see the applications in cell and molecular biology. So while using these animal cell culture techniques, you can use it for cell growth kinetics. Here, you can study the cells and their growth patterns. So, for example, what is the growth rate of a particular cell line? So, what is the rate at which it is growing in vitro? So, what is its viability at 24 hours, 48 hours, 72 hours? Such studies can be performed for cell growth kinetic studies. Next is the normal and abnormal cell division. Here, you can study your cells under various uh, chromosomal aberrations and chromosomal number that could be different for normal and abnormal cell division. And these studies can be then be used as an evidence for the mechanism behind normal and abnormal cell division. Next is the receptor studies and the signal transduction. All the cell-cell interactions are happening due to the interactions between various receptors onto the cells and the molecules which are passing on signal from one cell to another. So there are signal transduction cascades which are taking place in the cells and in the microenvironment of tissue. And these studies can be done with animal cell culture using different models in 2 dimensions and 3 dimensions using organoid cultures. All such type of studies can be done. Fourth is DNA replication and problems thereof. So you all know that there are number of diseases which can be caused by abnormal DNA replication. And such DNA replication abnormalities can turn into various diseases that cannot be cured unless and until they are happening at the localized level. So, in such cases, these therapies can be carried out to check the replication abnormalities of the DNA, and their problems can be dealt with various therapeutic applications of various signaling proteins, and these are, though at the research level, they are still in an infancy level. There are different techniques which have been evolved through gene therapy, and these can be performed in vitro cell cultures, and then the tissue which is developed from the repaired cells can be implanted into the uh, host, and such studies are underway to and has the huge potential for cell-based therapies. Next is protein synthesis and energy metabolism. Protein synthesis is one of the important properties of cells which is exploited by biopharmaceutical industry to develop the proteins of our interest. So, different therapeutic proteins can be obtained as a single cell protein or by purification techniques from the mixture of variety of proteins produced in the cell culture. They are being separated, isolated, purified, and they can even be further modified and used for therapeutic applications. The energy metabolism studies can be performed in cell cultures, and with the replicates of different number of cells, you can reproduce these results, which is otherwise doing these experiments in animals gets a very varied results and standard deviations. So these results can be obtained from the animal cell culture techniques because of reduced standard deviation. As your cells are the replicated cultures in small culture wells, these changes are minimum and you get the therapeutic protein and the energy metabolism studies in a reproducible manner. Next is a gene expression studies where we are looking at various drugs or various stimuli or environmental factors affecting the gene expression. Gene expression studies involve isolation of RNA, say tRNA, mRNA, and then their expression studies can be done by real-time PCR or the reverse transcription is done, and these DNA material derived from RNA can be stored for longer term for future study. So gene expression studies are very much important in the cellular mechanistic determination of action of any drug. So gene expression studies in animal cells is very much easy as compared to the techniques which are otherwise coming under various ethical issues for using directly into humans or in animals. Next study is apoptosis studies. Apoptosis studies are important in case of determining the anti-cancer activity of various drugs. When a drug is having ability to induce apoptosis in cancer cells is very much of importance as an anti-cancer agent, and hence the number of plethora of cancer producing uh, moieties as the carcinogenic material and their interaction with various drugs that can stop this carcinogenesis, and if the ones which can prevent or which can treat the cancer are important in producing apoptosis in cancer cells. And this different pathways that are involved in apoptosis generation or induction are studied in cell lines. Next is effect of external chemical agents and radiation can be studied in animal cell cultures, which provide a robust model where using animals or humans for such hazardous experiments for effect on the, the various tissues by these chemical agents and radiations studies are very much harmful for the livelihood. So instead of that, we can study these effects onto the cell lines. Next is a cell-cell and cell matrix interactions. As we discussed, each tissue is made up of a microenvironment that is specialized for that particular organ. And these cell-cell interactions and cell matrix interactions are specific for the specific tissue. These cell interactions and cell matrix interactions, they are affected in case of disease conditions. And to study the difference in all such interactions and cell matrix interactions in disease condition versus in healthy condition provides you with the insight of the disease which is causing various changes in the mechanism of the different functions taking place in the cell. So such studies can be done by using animal cell culture. Next is gene transfer studies. Now, inserting any new gene to a cell has become an important tool for studying gene transfer, their effects, and their expression to produce some another protein or the disease protein is replaced with a normal protein by using such gene transfer studies, for which cell culture models are best suitable, and hence the application of animal cell culture in various fields of, say, biopharmaceuticals are becoming more popular and essential. All the best for your future preparations for applications of cells in, say, cell and molecular biology techniques. Thank you.